Hello everyone, welcome to our channel Biological Concepts. In today's video, we will discuss on Frameshift Mutation, the definition, types, examples, diseases caused by Frameshift Mutation will be discussed here. So stay tuned. Now, first of all, we will discuss on what is Frameshift Mutation. A Frameshift mutation, also known as the framing error or reading frameshift. It is caused by the insertion or deletion of a number of nucleotides in a DNA sequence that is not divisible by 3. So, this frameshift mutation caused by the insertion or deletion of a number of nucleotides. Now, due to the triplet nature of gene expression by the codons, the insertion or deletion can change the reading frame, resulting in a completely different translation from the original. Now, what is codon? A codon is the sequence of 3 DNA or RNA nucleotide. 3 DNA or RNA nucleotide. Okay. That corresponds with a specific amino acids or stop signal during the protein synthesis. So this codon is a sequence of 3 DNA or RNA nucleotides. Now, what is reading frame? In molecular biology, reading frames are defined as the spans of DNA sequence between the start codon and the stop codon. So, due to the triplet nature of the gene expression by the codons, the insertion or deletion can change the reading frame, which results in a completely different translation from the original. The earlier in the sequence of the deletion or insertion occurs, the more the altered the protein. Now, let's discuss about the types of frameshift mutation. One is deletion mutation and the second one is the insertion mutation. So, first of all, discuss about the deletion mutation. In genetics, a deletion mutation is a type of mutation that involves removal of a portion of genetic material. So, when one or more nucleotides are removed from the DNA sequence, then deletion mutation occur. So, deletion means deleted, some portion will be deleted. So, here one or more nucleotides are removed or deleted from the DNA sequence, then deletion mutation can occur. This is the original DNA sequence and this one is the mutated version. Here the sequence is 5' ATGCGCTTAGA and in the mutated version this from the original DNA sequence this C is deleted, that is cytosine is deleted, and in the mutated DNA this C is not present. This C, that is cytosine, is not present. It is the 1, 2, 3, 4, 5, 6. The 6th number of nucleotide cytosine is deleted from the original DNA and as a result in this mutated version it will be ATGCGTTAGA 3'. Okay. Now let's discuss about the deletion mutation with example. This one is the normal or original version of DNA sequence, DNA and RNA sequence. Here it is the 5' to 3' DNA sequence, that is the sense strand, and it is the antisense strand, and it's the 5' to 3' mRNA sequence. The 5' to 3' DNA sequence is hydrogen bonded with a 3' to 5' DNA sequence. These nucleotides are hydrogen bonded, A pair with T by 2 hydrogen bond, G pair with C by 3 hydrogen bond. And look at this, the sequence of the 5' to 3' DNA sequence, that is the same strand of the DNA, and the 5' to 3' mRNA sequence is almost same, except this T is replaced with U in case of mRNA. And as we know that the codon is a sequence of 3 DNA or RNA nucleotides that corresponds with a specific amino acid or stop signal. Here, this codon, that is AUG, specifies methionine, ACU specifies threonine, CEA specifies glutamine, and USC specifies threonine, and so on. Just considering this portion. Next, this one is the mutated version. Why it is mutated? Because here a deletion of the adenine nucleotide. This adenine nucleotide is deleted here. Okay. So in the original version, the third amino acid was glutamine. After this mutation, this CAA is changed and it is CAU. The third codon is CAU. So it codes for histidine, whereas the CAA code for glutamine, it is code for histidine. And next one is the UAC. UAC code for threonine. But here the nucleotide AC, the third code nucleotide may be AC, A, T, GC, what, A, U, anything else. So it does not code for the threonine. It will code another amino acids. So this entire reading frame will be changed after this threonine amino acids. Okay. After that, this entire reading frame will be changed. Okay. So as there is deletion of the nucleotide adenine, so it is deletion type of mutation. Now let's discuss about the diseases that is caused by deletion mutation. Thalassemia is an example of deletion mutation in hemoglobin genes. It is caused by mutation in the HBB, HBA genes present on the chromosome 11 and chromosome 16 respectively. The unequal crossover within the globin gene complexes causes deletion, which in turn leads to backward shifting of the reading frame. Also note that this thalassemia is a group of blood disorder. And so thalassemia is an example of the disease that is caused by the deletion mutation. Now let's discuss about insertion mutation in genetics. Insertion mutation, also known as the gene insertion or DNA insertion. It is caused when one or more nucleotides are inserted into the DNA sequence. As there is insertion of one or more nucleotides, so it will be the insertion mutation. Look at this. This one is the normal DNA sequence and this one is the mutated version. Because here at that position there is addition of cytosine nucleotide. Okay. So after ATGCG, there is addition of one nucleotide. So that results in the mutation, DNA mutation. So it is insertion mutation due to the addition of cytosine nucleotide. Now let's discuss on insertion mutation with example. Again, this is the normal version. And this one is the mutated version. Here, cytosine nucleotide is addition of cytosine nucleotide at that position. Okay. In normal DNA sequence, it was ATG, ACT, CAA, TAC. Whereas in the mutated version, it is ATG, ACT, CAC, ATAC. So, as a result, when the mRNA is synthesized, the CAC, that will be changed. The CAA code for the CAA specifies the amino acid glutamine, whereas the CAC specifies the amino acid histidine. Okay, so after that there is also changed in the amino acid sequence. Okay, so this one is the glutamine. The third amino acid here is glutamine, whereas in the mutated version the third one is histidine. The fourth one here is threonine, whereas the fourth amino acid here is isoleucine. So the entire after that the entire reading frame is changed due to the insertion of this cytosine. So this is also this is insertion mutation, as there is insertion of cytosine nucleotide. Now, that is diseases that is caused by insertion mutation are examples of disorders that is brought out on by insertional mutation include myotonic dystrophy, cystic fibrosis, fragile X syndrome, etc. Thank you. If you like this video, please like, share and subscribe to this channel. Thank you.